You well enough?
Yes, I can.
Wonderful. All right, let's get started. Well, first off, thank you to everyone in the audience who is attending today. I'm quite excited to present the topic today on behalf of all our colleagues, collaborators, volunteers that have contributed to it. And I really do also want to say thank you to Anne Strong for the invitation to present, also to Paul Crump, who helped me sort through some technical difficulties earlier today to get this up and running for all of you to see. So as you can see, today I will be talking about Texas's botanical legacy. And I want to start by showing you this slide right now, which is a screenshot taken out of a very important publication that came out in 2020. So the talk today is really going to be about natural history collections, specifically about herbaria and the kinds of data that those specimens hold, and the kind of potential that they have for helping us understand the past, the present, and be able to answer questions and mitigate future realities. So this publication came out in 2020 from the U.S. National Academies of Sciences, Engineering, Medicine in Washington, D.C., published by the National Academies Press. The DOI link is there. I'm not sure how well I can share that with you, but since this presentation will be recorded and posted online, you'll have access to that afterwards. But what is a natural history collection? So a repository of non-living specimens and associated data that document the specimen's existence in space and time. So in this slide, you're seeing different examples of those. The small square that is in the bottom left, the photograph, that represents herbaria, preserved plant specimens, preserved in that way. And I'll show you, that's what I'll be talking about today. But the idea behind this publication, which is Biological Collections Ensuring Critical Research and Education for the 21st Century, was a plea and an outline for the value of these collections beyond what many people may think of them as antiquated processes and procedures of the past that have no impact or information to share with us as we look towards the future. And hopefully after today's presentation, you will agree with that, perhaps you already do, and also you will be aware of a resource that we have here in our state. So just to kick us off, specifically the natural history collections that we'll be talking about today are herbaria. And I'll get into a little bit more about what those are in the next slides. But just for you to know, there is a repository for herbarium collections that is available online to any one of you. I do repeat this link later on in the presentation, but it's called Index Herbariorum, and it's hosted by the New York Botanical Garden. This is actually, and apologies, the date is incorrect at the bottom of this slide, but this is from 2021. The title of the slide is correct. In 2021, Index Herbariorum reported over 3,500 active herbaria that contained 397 million specimens. They're around the world. There are more herbaria in the United States than any other country, with the next closest being the People's Republic of China, with about 375. To give you some context, the largest herbaria in the world, the top 5 at that time, are the ones listed here, with the Royal Botanical Gardens Kew at 8.1 million and the Missouri Botanical Garden at 6.8 million. Those numbers, of course, are higher now. But what is a herbarium? A herbarium is a collection of dried, preserved plants that are mounted systematically arranged for scientific research and education. You're looking at 4 photographs from the Phylogenetic Herbarium here at the Botanical Research Institute of Texas at the Fort Worth Botanic Garden in Fort Worth, Texas. So from the left, you can see the large scale. This is a view down our herbarium aisles where you can see a compactorized system containing metal cabinets, which are open in that second image. You can see a view of how these specimens are stored, a close-up of the kinds of folder organizations. You may find them in different herbaria. And then on the far right, Jessica Lane, one of our staff members here in the herbarium, who is working with specimens and arranging them out on one of our tables. Herbarium specimens do go back. I mentioned this, people think of this as an antiquated process, But, you know, they do go back to the 1500s. So the earliest herbaria are credited to an Italian physician named Luca Ghini at the University of Pisa. So about the 1500s, Luca Ghini was a physician teaching doctors, right? And at the time, medicine came majority from plants. And so an understanding of the plant world became really important. Luca Ghini had the desire to teach from direct observation. There were some books at the time, De Materia Medica coming to mind, by Dioscorides, to inform medical students, physicians, about what these plants look like and what their treatments, what treatments they might be used for. However, direct observation was something that Luca Ghini stressed for his students in order to better understand these plants and which ones you could use, and to give the students an idea of patterns across these plants and identifying them. So that's the 1500s. There's an image on the three-part image, which is on towards the bottom left of my screen. And that is an example of not Luca Ghini's herbarium, but a collaborator's herbarium, Francesco Petrolini, I believe. And this is, I think it's built about 1558, it's dated. It's named the Antibes Herbarium, and there's a publication link down there if any of you are interested in learning a little bit more about it. But as you can see, Luca Ghini, this idea of the hortus siccus, the dry garden, or the hortus hyemalis, the winter garden, the idea of preserving these plants. He wanted to teach his students direct observation, but these plants weren't all flowering all year, right? You had to deal with seasonality and winter when nothing was flowering. And so, well, few things were flowering. And so his idea was to take these plants when they were at their optimal time for observation and to find a way to preserve them so they could be observed in times when those plants were no longer accessible. Another way to share specimens. The herbaria that, it was not called an herbarium at the time, I think it wasn't until the 1600s that it got the name herbarium, but the hortus siccus was a bound collection, a bound herbarium, and that example you see in that three-part image is down at the bottom. You can see it was bound as a book. You've got plant specimens that are pressed and dried. You've got scientific names attributed to them, or you've got some names attributed to them. But it's not till the 1700s that we start to see that separation of those plants. Instead of in a bound book that you were limited by flipping back and forth, which was damaging to the specimens, you couldn't compare two things side by side. Linnaeus is credited with the idea of the single sheet herbarium. And this image on the right-hand side of the screen that is in black and white comes from a publication also credited down at the bottom. And you can see its similarity to the herbarium cabinet I showed you in the prior slide. Linnaeus wanted to be able to physically order these specimens in a way that reflected his understanding of the groupings he made. Hence that separate kind of cubbying system, which is this cabinet we still use this today. Here is an example of probably the majority of the specimens I'm really talking about today, where you have a archival, in this case it may no longer not have been archival, sheet of paper that has a preserved, pressed and dried plant specimen that is mounted or fixed in some way with glue, tape or string to that sheet of paper with an associated label. The parts are named on this specimen, so feel free to read through them if you wish, but in the anatomy of a specimen, really ultimately that primary label gives you the details about where that plant was collected, when it was collected, any other direct observations that were made about that plant when it was collected at the time of collection. And these are living, breathing entities in some way. Bear with me. So this is a source of primary data, right? So we don't have a time machine to take us back in time, but what we do have are these specimens that can allow us to go back in time. Imagine a past baseline of all these specimens collected back to the 1500s, that each of these plants in these specimens preserve a wealth of information about what environmental conditions those plants were growing under, right? As we know, the morphology, the shape of plants, the anatomy of plants is influenced by their environments. If we can take these specimens, which preserved those plants in the past at a moment in space and time, and study them, compare them across time, perhaps also across space, we can start to answer some ask some really interesting questions about how our plants are responding, have responded to past environments, how they are responding today, how they will respond in the future. So that scientific label with the dried and preserved specimen make this a scientific specimen. You'll see various of these, not as many as I thought I'd put in here, but you will see various of these specimens in the presentation. And just to give you some other idea and make sure the herbaria receive credit, the accession stamp, which in this specimen is featured in the top right-hand corner. It says SMU for Southern Methodist University. That is the herbarium that is the current owner and curator, caretaker, steward of this plant specimen. This specimen actually has a few other stamps on there, which show you kind of the trajectory of how that specimen moved around in space from the gypsum herbarium at one point at the University of California to SMU. There is a fragment pack on here because, as I mentioned, these specimens do have an active future. They were preserved in a moment in time, but they continue to be studied by researchers who are looking at species concepts. What is a species? And all of a sudden, I have all these 500 specimens plus of a single species in a herbarium. You know, imagine how much larger that becomes if you look at a global collection of herbaria of specimens, and you can start to notice differences in these plants, and it can help you sort out these species. So one of the other things you will see on these specimens are a bunch of notes, and we call them annotation labels, that can update information about this plant. Typically, it's an update about the scientific and the identity of the plant. There are also fragment packs in here because if you can imagine going back to the 1500s, the idea of being able to investigate, the molecular characteristics of a plant was not something that was not the purpose of the herbarium as Luca Ghini envisioned it, or perhaps he did, I'm not. But the idea that these specimens will be cared for in these herbaria and will survive there to be analyzed and looked at with whichever new technologies are developed. The fragment pack, the little packet that you see on there, often contains loose material, allowing researchers to take that for DNA extractions or allowing a place for them to put a dissected flower after it's been removed from the rest of the sheet. And I could talk ad nauseum about specimens in this way, but I will pop over to the next slide. So hopefully I've been talking a bit about the things that this slide is addressing already about the importance of these specimens, the fact that they are a plant from that past time and place that has been preserved, allowing you basically the equivalent of a time machine to go back and understand what that flora looked like and what those individual species were doing. There is an organization devoted to these natural history collections, and we affectionately call it SPINACH, which is the Society for the Preservation of Natural History Collections. It is a wonderful resource for any of you that is interested in natural history collections, and the website is up there. It specifically directs you to a page talking about why these collections matter. But these are among the areas in which these specimens can have impact: economy and trade. One of the things that we regularly do is identifications. And as you can imagine, specimens or plant materials moving across international borders, being able to identify them. We had a very interesting, you're not seeing a wood specimen on the screen, but we had a very interesting request of a loan of some of our wood collection, which is a xylarium, which has not been digitized. But the idea was to create a reference collection to characterize these woods, to allow some of our border facilities to be able to identify the materials that were coming in and out of the country. Changes over time, I have reflected, I've talked briefly about that. Environmental quality, the presence of a species that is sensitive to pollution, say a lichen specimen, at the presence in a certain place, but if it no longer exists in that place, gives us some indication of environmental quality. That's just one example. Of course, food and agriculture. As we know, we have been modifying wild species to these crop crops that we're using today, having an understanding of how all of these are related gives us information to improve our crops and perhaps to improve our crops. Public health and safety. This is another aspect, national security, invasive species. There are different tools today that are complementary to these physical herbarium specimens, but no other that represent a primary source where you could actually still sample that material with a new technology. And I will talk about one of those specifically, which I'm sure many of you know about, which is iNaturalist. And scientific treasures, of course, and this specimen on the screen right now, those of you that may have been squinting, hopefully these next images will make it a little bit easier for you to see that label. So this was a specimen discovered in our herbarium on a past digitization project. And if you are reading after the word legit, which refers to the collector, you are seeing the name John Muir, and you are seeing 1875 Sierra Nevada, California. So this is in fact the John Muir, who is the father of the National Park System here in the United States, the Scots American, and a wonderful treasure that we discovered in our collection that is digitized. It is available, it is online, and all of you will have a chance to search for it. What often becomes very important as you're looking at these specimens are the barcodes, the unique identifier that have been applied to these sheets at these collections. That's often one way to look these specimens up. But of course, my email address is at the end. So if anyone's interested in accessing any of the specimen images or images I show you, I'm happy to share those after the fact. All right, all of that to say that there is a community of natural history collections globally. There are herbarium repositories and aggregators, and there are Herbaria consortia around North America, around the world, of course. But specifically, I'd like to draw your attention to this one, which we call TORCH, which stands for the Texas Oklahoma Regional Consortium of Herbaria. This is a consortium that was developed, or that was founded in 2006 by Amanda Neal at the Botanical Research Institute of Texas, Wayne Ellison's at the University of Oklahoma, and Tom Wendt at the University of Texas at Austin. And the intent of this organization was to advocate for collections in our two-state region, to share information, to engage students, and to really help the larger and wider community find the value in our collections. That website is up here and it's on the screen. We're in the process of making updates to the website. So if there's something you're looking for and don't find there, please don't hesitate to send me an email. There's also an email distribution list, which will be posted soon there, that any of you can choose to subscribe to if you're interested in learning more about this project. But this project, it's an open membership. Anyone can join who is at a herbarium, who supports an herbarium, who wants to learn more about herbaria. And that image that you're seeing comes from the bottommost image is a photograph of our most recent annual meeting, which took place at the University of Oklahoma, hosted and put on by Abigail Moore and Amy Buteau. And you can see a lot of wonderful students and professionals that are coming together to present. There's information about this meeting, and the program is available on that website if anyone is interested in seeing. And if you're interested in attending, just wait till next year. We haven't decided on a meeting time. All right. The reason I showed you the information about that consortium is to set the stage for, there are all these specimens, they're distributed in these collections around our two-state region. And but we do have a group. We have a group that is here to advocate for, that is poised to kind of take on any initiatives or projects. So now let's go over to kind of the problem that we're thinking and dealing about, the larger scale problem. So in our two-state region in Texas and Oklahoma, that represents 11% of the contiguous United States. So that contains 17 level 3 ecoregions as defined by the EPA, by the Environmental Protection Agency. It connects mesic forests, which are in the east, the southeast, to the interior arid grasslands and deserts, which you find in the west, which makes for a really interesting confluence of those two. It links the temperate and the subtropical biomes, another crossroads, and hence we call this the American crossroads of ecological and biological diversity. And that phrase happens to be the title of the grant, which I will talk about a little further here. but a little bit more. Some of this you've already heard, but it helps me to set the stage of why the diversity is so high, the plant diversity is so high in our region. So these are 3 maps taken from the references listed under the title for the map. We've got precipitation, and here in our 2-state region, you can see a move from east to west of a mean annual precipitation variant from greater than 150 centimeters a year to less than 12.5 centimeters. So, a change of 1.5 centimeters every 10 kilometers, which is a phenomenally extreme moisture gradient. You're seeing elevational changes from up in the, mostly that tilt from sea level to 1,300 meters with the Ozarks and the Trans-Pecos, where you've got outjuts and outcrops there, isolating populations of plants, encouraging diversity. And then you've got temperature from north to south. You're seeing that mean January temperature ranging from 0 degrees Celsius to 18 degrees Celsius and traveling through 5 plant hardiness zones. That's not been updated. So you're seeing this incredible diversity of environmental gradients. You're also seeing this incredible diversity of geology and soils, surface exposure being shown on the map on the left from Holocene alluvial deposits to 1.6 billion year old outcrops. And then you've got on the right-hand side a map showing you that we have over 100 soil types, major soil types in our 2-state region. So all of this lending itself to this idea that we have an incredibly diverse botanical heritage in this area. If you look at our 2-state region, 31% of all the native species in North America, north of Mexico, are encompassed in this region, which represents 4,945 native vascular plant species and 213 families. Oh, cannot spell the word most, but in the next point, you'll see Texas is the second most diverse state in vascular plants. We've got 325 in Texas alone, plant taxa, restricted to the state. And then we're looking at the richest centers of plant diversity being the Ozark Highlands, the Big Thicket, the Edwards Plateau, and the Trans-Pecos. The map that you're looking at on the right comes from the Synthesis of the North American Flora. This is BONAP. The website is listed here. And this shows you the density gradient of these native vascular plants. And you can see roughly on that scale there what kind of density we're looking at. This map here has been taken from NatureServe. So these are in this region that has this high environmental, all these extreme environmental gradients, this crossroads. We're also seeing risks, right? As we are seeing in many places, risks of overgrazing, oil and gas extraction, exotic and invasive organisms, fire suppression, pollution, hydrological changes, urbanization. And in this range, you can see that there are 2 globally ranked hotspots. We sort of see the North American coastal plain represented by our Gulf Coast, the Mediterranean pine oak woodlands represented by our Trans-Pecos area. What this map here is showing you, and I've taken this text from the smaller text that you cannot easily read underneath the map of the United States. It shows the concentration of limited range species and also highlights those areas where the species composition differs from those adjacent areas. So really, this is the idea behind this map, is it's highlighting these areas of irreplaceable locations. These are opportunities for conservation. And you can see all those areas that are highlighted in our 2-state region. So we've got a wealth of data. We've got all these reasons that our area represents really interesting and an interesting place to be able to ask some of these questions about what causes this diversity, and what will happen to this diversity under changing future climate regimes. So step into the next thing is a National Science Foundation grant. So this is a project that was put forth by the Torch Consortium, which we talked about already. So although the name of the grant is actually the American Crossroads Digitizing the Vascular Flora of the South Central United States, you will often hear this grant called the Torch Grant, the Torch TCN, or sometimes American Crossroads. TCN comes from an idea, the Thematic Collections Network. This was a program that the National Science Foundation started 11, 12 years ago or so to fund the digitization of these biological collections, because they thought that these were so important to helping us understand our environments and changes we're facing. So this grant that we were successfully awarded in 2019 was to digitize almost 2 million specimens, 1.7 million from about 41 herbaria within the Torch region, 0.3 million from 5 additional herbaria outside the Torch region. And you may ask, well, why do you include herbaria outside the region? Well, because of course, this is a distributed network of specimens. Texas specimens exist in herbaria outside of the Texas and Oklahoma region. But interestingly, some of the earliest collections we have in Texas were collected before there was an established herbarium in our state to house those collections. So those specimens exist at other places like Harvard, like the New York Botanical Garden, like the Missouri Botanical Garden. And then, of course, there are other collections outside of our region that have much newer concentrations on Texas collecting, which, for example, the University of Kansas, University of Arkansas. The way this grant is structured is that there are 5 leads: the Botanical Research Institute of Texas, represented by PI Principal Investigator Peter Fritsch; University of Oklahoma, Abigail Moore and Bruce Hoagland; the Oklahoma State University, Mark Fishbein; Texas A&M, Daniel Spalink; University of Texas at Austin, George Yaskevich, and it's a collaborative award. So down at the bottom, you'll see NSF, National Science Foundation, credited with those multiple award numbers, so you can see that total. But this is a much larger grant than just those 5 leads. They happen to be the leads to which subawards and provider collections report. So if you are interested in knowing which collections are being digitized, you can look at the names that are up on this screen. The different colors represent the way we've grouped these collections because of logistics, because of physical proximity, and perhaps other reasons. But really, these are the goals of the grant. We want to disseminate digitized data through an integrated symbiota platform, and I'll talk about what that is in a little bit. We want to develop and implement innovative strategies to increase this digitization workflow efficiency. And we want to recruit and engage students and citizen scientists in project-based broader impact activities. I'll get to some of what this means. But just to tell all of you, so these are herbarium specimens we've been talking about. They live in these cabinets. I've showed you multiple examples from the 1700s up till, what was that taken, 2021. But they're locked in cabinets, and that's the whole goal behind this grant. If we can take these specimens, digitize them in such a way that they become now available for computational analysis and just accessible to everyone who doesn't have the means to physically get to an herbarium or to all herbaria, digitization becomes a very important tool for access and for the idea of consilience, right? Taking us back to E.O. Wilson, the idea of bringing us back to this understanding of our environments as this dynamic, moving parts that have multiple influences. So digitizing these specimens stages them for integration with other systems. But it all starts with how we digitize these plants. So for us, it's generally been a 3-part process. You're looking at sort of 3 kind of separated images. On the far left, at least on my screen, you are looking at a digital single lens reflex camera with a light box which isn't currently lit. Below it, you're seeing an image capture through some Canon software. It happens to be the camera in this system. So that's the image capture. Second stage here is you're looking at an example of a label. I'm sorry, it's not from Texas, but this image shows you a typical herbarium specimen label and the different elements that are on there. So that second stage, transcription, taking label data off of specimens, becomes critical, because with a bunch of images, what does that help us do if we can't discover those images? So this transcription process is a huge undertaking and rather important to both faithfully take the information off these lists and parse them into the appropriate fields so that you can search for all the plants from Lubbock or whatever you wish to do. The third stage of digitization is the idea that each of these specimens has, if not a coordinate, a geographic coordinate because they were collected recently, they all contain textual information about where these plants were collected. This is the current phase that, at least in our Brit lead area, we find ourselves concentrating on, which we call georeferencing. The idea that you have a textual string saying where this plant was collected, and you have to turn that into a geographic coordinate that can be placed on a map, with, most importantly, an associated uncertainty that encompasses sort of that uncertainty about where exactly that point was. If it says it was a mile south of a certain city, well, how has that city changed over time since it was collected? Or has that road diverged? So I think that hopefully you can see that georeferencing is often as much an art as it is a science. And it's actually one of the really rewarding areas. And I will talk very briefly about this, but we do have other presentations I can share with you if you send me an email about this. All right, so 3 parts: imaging, transcription of label data, and then the georeferencing of the collection locality. Something rather interesting we found, because some herbaria have this workflow modified slightly, but we found that if we can invest in that first step, imaging these specimens, all the downstream digitization efforts can happen with a digital image. Now, that becomes, some of you are probably already thinking this way, but that becomes extremely important when we're talking about things like COVID. When the pandemic shut us all down, we were able to keep our staff and our students employed and working from home because they had access to this large mass of images of specimens and were able to work off of those. If you'd like to see this in a more detailed format, that digitization workflow, it's listed here. The part in blue is the part that wasn't really addressed in that prior screen, but this addresses sort of that digital sustainability of these specimens and what actually happens to those images of the specimens as they move forward. We talked about the image capture of the specimens, which is encompassed by this green box, which talks a little bit about how maybe all you capture with an image is a scientific name of the plant, and you wait till a later point in time to get all the rest of the data out. But that image has to go through various processing stages, and it has to be uploaded to a site that it has to be backed up locally, of course, and it has to be put on a site that can serve can be a web server to serve images so that the public and everyone and researchers can access them. And that's a bit about what sort of, well, part of what the central section talks about. And then, of course, we talked about the complete transcription and parsing. There are 2 programs that are named there, and I'm afraid I didn't include the direct links to URLs to those, but I'm happy to provide them afterwards. Notes from Nature, which is a wonderful crowdsourcing virtual platform to allow people to transcribe specimen labels from images online. Biota, which we will talk a little bit more about, but this is the system, this is a software platform out of which all these specimen data are served for our grant. And then georeferencing, geolocate is another platform that we use to georeference these specimens. All right, so this is really what I'd like you to sort of take home, and this is the resource that I feel obligated to make sure you're informed about so that you can access these specimens as they're digitized. One caveat I will say, so the date at the bottom of this slide is correct. So these are some numbers that I pulled just yesterday, but because of that, some of those more complicated slides earlier about our digitization workflow, specimens change by the minute as they're contributed by different herbarium collections to this online website, which we call the Torch portal. And it's the URL is there. It's portal.torc herbaria.org. The QR code on the top of the screen links you to that database. So this is a database that's accessible to all of you. If you are sharp-eyed and looking at that screen capture on the bottom of that website, you'll see that I'm signed in. You can create an account, a free account. It doesn't really give you, well, it does give you some tools you can use to comment on specimens and to communicate with the collections that are disseminating these specimens. And so on this portal, thus far, so as of yesterday, we have over 1.1 million, what we call specimen occurrence records, so specimen records collected in just Texas. 897,855 of those specimens have images. So slowly we are putting images, associating them with specimen data online. Slowly we are fleshing out specimen data. So if what you see is not online, check again in a day, in a week, or contact the curator of that collection to find out more information. So there's 41 torch herbaria on that screen when you first come to it, but there are the others that we mentioned that are outside of this torch network that are kind of nested that you're also searching. This database is searchable by scientific or common name, by locality data, by collector name and number and date. But another reminder that if a collection is only represented by an image and a taxonomic name, if you type in locality information, it won't pull that specimen up. It doesn't mean it's not there. It just doesn't mean the data. It just means that the data may not yet exist to find it through that means. Part of this grant is integrating with larger aggregators and initiatives. So we report out, and I'm sorry, I'm going to go back a screen. This platform, the Torch portal, is built off the Symbiota software. So that's that word I keep repeating. The Symbiota platform can push its data to our national hub, which is IDIG-Bio, standing for Integrated Digitized Bio Collections. I just popped in there this morning to check, and you've got a significant number of specimen records. Now, this is our national hub that aggregates data not just from plants, but other organisms, other bio collections. So I encourage you to pop over to that URL, which I posted, and investigate things there. I will tell you that our Torch grant is focusing on getting data into that Torch portal. So that probably represents the best place for you to go and pull Torch Texas data right now from herbarium specimens anyway. And then secondly, on this screen is the GBIF, which is the Global Biodiversity Information Facility. This is a global aggregator of biodiversity data, including aggregating data such as from observational platforms like iNaturalist. And that would be an end goal for our data to propagate through these systems so they are fully disseminated and interoperable. All right, so I'm running relatively short on time. So I have a few quick examples that I may pop through. This URL will should take you to the search results for a quick search for Tarrant County specimens online. So when I checked this, and this would have been 6 to 8 months ago, apologies, I didn't put a date on there, but this was, I found that there were 19,000 specimens, over 19,000 collected in Tarrant County. Over 15,000 of them had images. The oldest specimen that was collected in that area was 1876. For those of you that are history buffs, this is an incredible tool to kind of align your interests in history and explorers in Texas. The most recent specimen is updated. It was actually 2024, but in this case, it was 2023. And that represents collections across 90 different herbaria. So you can start to see that integrating all these digitized collections from many herbaria is the only thing that gives us that big view of these species and where and when they occur. Here's one of those recent collections, just an example. I did a search for things flowering in January, and of course, dandelion is one of the things that popped up. So this is from the University of Texas, El Paso Herbarium. I'll pop to the next screen. I mentioned that the oldest specimen in that Tarrant County data set was 1876. It was collected by Julian Reverchon, and there is a photograph of Asclepias stenophila, which is what this specimen is on the left, from iNaturalist, from a user, Bendira 44 John, from October 2023. So you can see what it actually looked like. And that leads me into this slide, which really talks about the complementarity of iNaturalist, which I'm sure many of you are familiar with, and these herbarium specimens. It presents a really great way to look at the distribution of these species. Ultimately, the one thing to note is that herbarium vouchers represent sort of represent a primary source, which means you can go to the physical specimen. You could, if they're not glued down, move those leaves up and down. You could open up a flower and look at the rostellum to see what it looks like, because maybe that differentiates species, and that's something you cannot see on a photograph. However, the speed, the crowdsourcing that you can get out of iNaturalist make it such a powerful tool. So between these two tools, one of the things we've regularly done is we've taken, say, for a county of Texas, we have taken a list of all the herbarium vouchers from that are known or digitized as yet from that county. We've taken a list of all the observations in INAT from that same county, and we've compared those two lists of species, standardized them, so we're talking apples to apples, and we're able to see, well, oh, these are new observations in iNaturalist that have never been vouchered, and is it important to voucher them? So the these digitized data allow us to identify gaps. It also puts the power in the observer, in the general public. Communicate with your local herbarium if you think you've found something interesting, new, something that represents a connection between disjunct populations. Here's a wonderful case study. So this is Dr. Russell Pfau, Tarleton State University. And these slides come from Kimberly Sasson, who worked on this project, and Ashley Bales, who helped compose the slides. But we're looking at Anemone caroliniana over here, which is also the subject of what Dr. Russell Pfau has been working on. Kimberly got rather interested in working on this project with Dr. Pfau and generated some of these comparisons to tell the species apart, as you can see in that photograph on the left. This map represents, at the time that these were generated, the number of INAT observations that existed of the species. You can see in 2018, it went from 4 observations down to 2023, there were 150 observations with 86 observers, and you can look at that kind of distribution. So the power of this tool is absolutely phenomenal, but you can now see that iNaturalist shows that there's gaps in our specimen records. So today we see an Elliott collection from the Academy of Natural Sciences in Philadelphia, collected in 1814. We see a Linheimer in 1843 in Texas. We see a Reverchon collection from 1878, and that now sits in an herbarium and sits online digitized with a new specimen collected by Kimberly Sasson. So I will say that we do need all of you to help us find these gaps, to fill these gaps. I will say, make sure that you are communicating with an herbarium to find out permission for collecting these specimens, procedures for collecting them, and all that good stuff. I'm happy to talk to you or share more information about that in a different presentation. I skipped over that slide because I'm running short on time. So I'll tell you, before georeferencing, that third step I told you about, we're looking at, in the Brit collection only, the distribution of Anemone caroliniana. After being able to georeference those legacy collections that existed from the past, we're able to see a much more realistic distribution of these plants. All right, I'm getting close on time. But this is a slide that comes from a publication called the Extended Specimen Network, a strategy to enhance U.S. biodiversity collections, promote research and education. It came out in 2020, and it took this idea of the extended specimen from also an earlier article, but it presented it in such a way that you're looking at it in terms of a herbarium specimen. You have the original object, that field collection. You have all the associated images. You can create these morphological data, ecological, environmental samples, and you can see these different extensions from the original object to the primary, secondary, and tertiary extensions. But it all comes, and it's all based off that original physical collection, maybe that nested sampling, but those physical collections. So the impact, digitizing these physical collections in a standard way according to internationally accepted metadata standards, means that you can now start to integrate these with other data sources to be better informed. I have two slides, Anna. Okay, so I did want to slowly get here to the end to concentrate on people, because this has been what has made the project, what has made it happen, and what has made it even more enriching for those of us that are actively working on it. So staff, students, volunteers have assisted with this project, funded and unfunded on this grant. If I look at the contributions through one of those websites I mentioned to you, Notes from Nature, which was an online crowdsourcing transcription platform from specimen images, we have had, and this was, we have had over 3,300 separate individuals assisting us with digitizing specimens collected in Texas and Oklahoma. And actually, that 3,300 is actually probably just reflects, since we posted mostly Texas things, that's just Texas. So aside from that, we've also had institutional volunteers that have helped us do this. We have a program here at the garden called the Armchair Botanist Program. We have a presentation tomorrow, so hop over to our FWBG website and do a search for Armchair Botanist, and you'll find a new presentation coming up tomorrow. But many of the individuals, there are many groups that have volunteered, individuals and groups that have volunteered with all our herbaria, but I'd like to do a quick shout out especially, and perhaps this is in particular to the collections that we've worked on, but the Texas Master Naturalists and the Native Plant Societies for both our states have been really phenomenal in supporting the digitization efforts by direct digitization, by sharing, by advocacy for our collections. And then lastly, I want to mention on this slide that this project did fund 20 students attending, 20 students over a 10-week summer internship where they were required to utilize digitized data to contribute to digitizing data. This picture on the top represents them at our annual conference presenting. There's a short URL down there if you'd like to see the kinds of topics, because a lot of them have to do with climate change and changing phenological patterns in some of these plants. And then, of course, nothing's real until you take them out in a field and you have to collect your own specimen, as Abigail Moore is doing down there at the bottom, showing a student. Ways to get involved: reach out to your local herbarium for filling in the gaps and for direct digitization. Email, and of course, if that doesn't show fruit, or there's not a way, or you're physically not able to help them there, you're certainly welcome to email us at the Phyllocology Herbarium. It's herbarium@fwbg.org, and you can reach us that way for getting engaged. And then my last slide is just a big thank you, because this has been such an enriching project to work with so many people, and the power that we have now with all these digitized records online because of what y'all have done becomes pretty important. And those of you that are new to this project, pop over to that portal. Sorry, I didn't put that link there again, but pop over to the portal and start to ask some questions, and certainly, you know, punt them our way, or let us know if there's something you'd like to see. Of course, iDigBio, because they have supported us, it's the National Hub supported by National Science Foundation. They have supported us and our efforts at the National Science Foundation. And lastly, my email address is there, so feel free to pop me a message anytime. And Anna, I think I'm done, and I've left us about 10 minutes for questions.
Yay, thank you very much, Tiana. So there are some questions. There's one from Brooke Best who says, what do you think the red circle is around Lubbock in the Nature Serve map that you showed? It was a very large red circle. It was, I don't know, your fourth slide, maybe.
Yep, you're right. Hold on. I know there's a faster way to do this, but I'm afraid of
that's okay.
Messing with your. So Lubbock and that area, I honestly don't actually know what that's a reflection on, but Anna, that might be a really good question for you. Why is that there? Are we seeing, I mean, we're seeing some of the peaks up there, right? So we don't have a topographical map in here, but I can't exactly place where that is. But what do you think?
That's, well, it's around Lubbock. I mean, I'm going to guess oil and gas, but I don't know what other threats are around Lubbock. So if anybody has any ideas, you can submit your ideas as a question. That's or let me know through another means. Another Chevy Tang asked, or just says, this is impressive, and the data access is great. It's really helpful. Lisa Egner asks, are there similar databases such as BRIT that covers other parts of the US? And I posted the Torch portal and said Torch can be searched for anywhere actually outside of Texas. Do you have any other things you would like to say about that?
No, but, and I apologize for not remembering who did it, but hopefully you're now looking at the Torch data portal right here. So this is one way to access all these data. If I go to the search collections, you're here able to see, I'm going to minimize this for a second, but these are the different networks that consortia, I mentioned Herbarium Consortia earlier. These are the different consortia that are sharing data here in this symbiotic system. If I expand Torch, you'll see some of the herbaria I listed earlier, but you can see that there's also Arizona, New Mexico, Canadian herbaria, Midwest herbaria, a lot of those. But I will tell you that the iDigBio portal is extremely useful. Sorry, I don't have it. I know I have it bookmarked somewhere, but this is a really good, useful place to go. I will tell you again, just that if you're specifically looking for Texas, Oklahoma data, that Torch portal will be the best place to go. But iDigBio is our national aggregator, so that's another good place to go.
Beth Anderson says Torch Herbaria presents images in medium large format. The large format images are really helpful in seeing broad details, but may not always show fine details of the sort you'd use a loop to see. For example, glands or tiny hairs. Are there even larger scans stored somewhere that can show these details, or do people come into the herbarium to see them, assuming they even persist through time and can still be seen?
So that's a really good question. So it depends. So different herbaria are imaging with slightly different systems, and there are choices that each herbarium makes about what kind of web derivatives they share. So it is possible that there are larger images that are accessible. And in that case, I would encourage you to email. If you go to any one of the specimens, and I can't think of a name to search that will return a short string right now, but if you go to any one of those specimens, you should be able to see down at the bottom who to report questions or comments to. There should be an email address, and you can certainly email that individual directly to ask about those higher res images. Depending on the capacity of the herbarium, you may also be able to reach out to them directly and say, can you take a microscope image of this or that for me? But you bring up a really good point, which is that these digitized specimen images are surrogates for the actual thing. They can be very useful for some things, but there are limitations to them. For some taxa, you're right, you can actually make identifications from those images, but for others, you're simply not able to do so.
Changing gears a little bit, another question is there fungi data available?
There is. So there is, sorry, let me do escape. That's the wrong thing. There is another, well, you are probably aware of, hold on, let me go home. So this is built off that same Symbiota software. This is for mycological specimens, and it works similarly to the Torch portal in that you can see various herbaria. So here are Brit collections. Let's see, you can see the geographical distribution, but works the same way as the Torch portal, and you can do searches in here. So yes, the answer is yes.
Can iNaturalist work on bryophytes too?
I have had iNaturalist return identifications to me when I submit images on iNaturalist. There may be a better person to answer this. There are, of course, microscopic characters and other chemical analyses that may be, well, maybe not so much for bryos, but yes. But as far as how accurate they are is a whole nother story.
Are lichens well represented in dried herbaria in Texas and Oklahoma? And are there resources in Texas available to run genetics to properly identify them?
There are, so different institutions, you'd have to contact them individually to discuss that, to figure that out. We do have a molecular lab, for example, here at BRIT, we do have a lichenologist, Manuela Dal Forno. And so she does use our lab to characterize some of these lichens. Lichen specimens can also be found online in a Symbiota portal. And this is, I just pulled up that website for you here. Again, it's built on a Symbiota platform. And it just changed. It's really, they've just updated and upgraded something, so I can't navigate here. So I'll let you, I won't navigate you through here, but there is a lichen portal where you can access preserved specimen data.
And as an add-on to that, sorry, were you done?
Done enough.
Yes, okay. Don't mean to cut you off there. Is iNaturalist the primary resource for lichens in Texas?
I suppose I might need a little more clarification about what you mean.
I think it was an add-on to that previous question. And so I think that you already answered the question.
Okay,
that,
yeah.
No, that this portal is probably a better place to go, maybe.
Yeah, although you know that so many of the value of linking, what I've started to do with personal collections is making an observation in iNaturalist that I link to that specimen occurrence record when it gets deposited in our herbarium, because the value of seeing those field images and linking those to the physical preserved specimen, I mean, is huge. And depending on certain organisms like lichens, seeing the coloration that you have in the field and seeing, you know, depending on how that changes over time could be huge. So, yeah.
We have a couple people that are saying very nice things about you, just in case you would like to go back and look.
I see somebody call me, thank you, David, a rock star, which comes right after the lichen comment. So that's pretty awesome. I thought that was appropriate and funny.
Are there examples listed of how organizations have utilized the database for specific questions?
That is such a good question, Liz, and it is an entire other presentation to talk about that. I suppose for specific examples, there are papers. There's a paper that if you email me, I can share with you talking about novel uses for herbarium specimens and how they've been used in certain studies. But it often goes back to these larger research, publications that are coming out and what they credit and how they're crediting those collections. Something that digitizing these specimens has opened up is the idea of attributing them to the collections that digitize them, to the individuals that collected them. I encourage you to look up Bionomia as one resource, BIONOMIA. Well, I guess I could pull it up. Well, since I don't know the URL and I don't want to waste our time right now, I won't. But Bionomia is something interesting to look at. And so just email me, and I'm certainly happy to send you some publications if you'd like to see them.
Just curious, how does the torch system determine what common name to include in the archival database for a given species?
Marcia, that's an excellent question. I don't actually have the answer to it. So I'm afraid maybe ITIS. I'm afraid I don't know. I'm afraid I don't have the answer to that. I rarely search for that common name. So I'll often, if I only remember a common name for something, I'll look up the scientific name and pop that back into the torch portal. One of the interesting things that portal does do is if species has gone under different names, it will search all those different names it's gone under. Under two of the search result view, you can view it as images or as a table. On one of them, I think the images, you can actually see which other scientific names that are thought to be synonyms it's also searching.
And just in case you didn't see this, Mary says, I actually entered some Texas and Oklahoma herbarium collections from Mobot during the COVID lockdown. So great project.
Mary, that's spectacular. I can't tell you how much it means to us, because there's no way any of us would be where we are without the help that y'all have given us. So thank you so much.
And I think that's all of the questions. So since we have 1 minute, unless you have some other general comments, I think we could try to wrap this up.